I'll start. My name is Mikhail Rosashansky. I am a electrical design engineer at Deep Sea Power and Light, and I am here on this expedition testing out our new MXDC cam on Little Herc. So all the footage that is going to be gathered during this dive on the main camera is using that camera, and it's a really nice, high quality imaging platform that we've developed with a collaboration with MBARI, which is the Monterey Bay Aquarium Research Institute. Let me see here. I think I'm excited for...
Go for that stalk on the right.
Can it be more than one thing, or does it have to be one?
It can be more than one thing.
Okay.
Is one of them a burrito?
Yes, that. So one is food. One is food. San Diego has a lot of food diversity and culinary things going on, so I'm very excited to eat like a nice taco and some ceviche. Although I might find that even on the island here.
Yeah,
so.
ceviche for sure.
And then I'm also excited to see my partner and our two cats.
Yeah, we got it anyway. It's
cool. Yeah.
Crinoid.
I bet they'll be happy to see
we wouldn't
you
mind
too.
having a go at
Yeah,
that one now.
I think so too.
Is more settled on that? That's another crinoid, so don't worry, we'll keep going. Can you swing to the port side?
Yeah, I've got to get over that way anyways.
What are your cats' names?
Sweet one.
Oh.
So yeah, let's go for that one there.
So we have a 1-year-old. His name is Harrison, like the Beatles.
Quick zoom on the fly.
Harrison from the Beatles, and then a 2-year-old called Mimi.
Urchins, eh?
Nice, I got it.
Okay.
I have a cat named Mimi.
Oh, you do?
Yeah.
Nice.
I do. Mimi and Kiki.
Oh, that's cool.
But Mimi's full name is Marcella.
Oh, okay. Ours is just Mimi.
This is just Mimi.
Yeah, it's not short for anything.
She's only Marcella when she's in trouble.
I love that name, Marcella. It's a great name. Marcella.
How did you get Marcella?
Have you ever seen Marcel the snail?
No.
It had a viral moment in like 2009. It's like this little snail that talks like this. But there was a movie and it's a feel-good movie for me.
Right.
Is it like stop motion?
Yeah.
Yeah.
And it's oh my gosh, now I can't remember her name.
Zoom on the fly again. I'll just keep getting a bit closer.
Stalk there. Huh.
Don't know what that is.
Cool.
Might be a xeno of some kind.
It's Marcel the shell with shoes on, and it's voiced by Jenny Slate. So it's really, really funny, but Marcel's real name is Marcella.
Nice.
If you haven't watched it, 10 out of 10.
List, yeah.
Okay, kill it.
A little Holothurian there.
Let's keep going. So really irregular nodules now again. I was saying that happens from time to time on this traverse. There's another Holothurian there. Keep going.
I think I've got a vertical dropout again.
Oh.
No worries.
Yes, I do.
John, we're not too far from your home. What are you most excited about when you get back?
That snail looks like that little visual that you just saw.
Yeah.
That looks kind of freaky.
No, it's cute. It's cute.
Wait, let me look that up.
And I'm really looking forward to getting home, having a drink, sitting with my wife, looking at the view, all those sorts of things.
Like you may be back.
We've got a very busy end to this year. There's another research cruise coming up shortly. And last time I counted 3 conferences.
Oh,
yeah.
In the next like 2 months?
Yeah.
Wow.
Yeah. As many as possible we try and do virtually. But yeah.
That's a lot.
Within the team there's lots of stuff happening. Yeah, the whole year has been a bit like that. But, so I guess I'm looking forward to Christmas.
Yeah.
Nice.
just to wind down and chill. Yeah. John is our watch leader. He lives in the Cook Islands. And he is a geologist, so we have had a geology lesson every watch.
Let's
We
zoom
sure
on that.
have.
So hopefully I
They've
can
been
give you another
wonderfully
one
educational.
later today. 'Cause this particular
So-
traverse is trying to look for an area with less nodules in it. So far we haven't been terribly successful at that. But it's not unexpected. When we planned the traverse, we knew that the beginning of it would have a lot of nodules, just a question of when it drops off.
And we launched at 8 o'clock, 9 o'clock last night?
10 o'clock.
So 10 o'clock. So
Yep.
we are 14 hours in, so 14 hours and no drop off.
Pretty good.
Yeah.
And we're going for a 36-hour dive, so about just over a third of the way through L1010. Sandy, what are you most excited about?
I am excited about a lot of things when I get home. I'm excited.
Run
Well, I'm not
and
going to be
zoom.
home for another month,
Oh,
but I'm excited to just be back in my home. I'm excited to go for runs again. I'm excited to go to the beach. I guess I can go to the beach on the
Okay,
Cook Islands
zoom
too,
there.
but let's see. Very nice.
thank you.
They have all of their
Thanks,
little
Jana.
markets coming back into season now because it's in the fall time. So I'm very excited to go because St. Pete, it's really pretty and decorated for the holidays, and they have all their little fall markets, their farmers markets, different events. And so I'm excited to go and just enjoy the city atmosphere again.
Sounds nice. Holiday markets are so fun.
They are. Oh yeah, I'm Sandy, and I'm data logger on the Nautilus, and I'm very happy to be here. Marine biology is my background, and I'm at University of South Florida.
And sitting in front of you, we have another University of South Florida association, or an associate, an associate with the university, Matt. Matt, are you excited to get home?
Yes. And with profound apologies
You good with that
to
thrust, Ray? Do you want to zoom in on that?
all family and friends back there, I think my answer is salad. Very,
I
very
feel
excited
that.
for some salad.
Yeah.
And Can you zoom there, Jana? And my own shower.
That
will
would
also
be nice too, yeah. Looks like a little shell. Oh, that is so
Yeah,
cute.
it's a gastropod or something.
Yeah.
And you got the one on the left, another one of those tetrapody things, and a gastropod. Can we get a doubler on that one?
That is so cute.
You can see the tentacles coming out of it. There we go.
Oh, the controls are little fine there.
We'll try and get a good photo of this one for our catalogue, I think.
Nice.
Nice. And zoom out, please.
That's
Perfect.
Marcel, the
Thank you.
shell with the shoes on.
Okay.
And if you've been tuning into our watch, Matt is our navigator. He is also the keeper of time and space.
Our resident Time Lord.
Yeah, Time Lord.
When I'm back home, yes, I do work at the University of South Florida, namely our Center for Ocean Mapping and Innovative
Too late for that?
Technologies we have with
I
NOAA
Oh,
Office
uh,
Coast
That might
Survey.
be a little, we can give her a try. Oh, that's another one of
Yeah,
those things that we
let's
didn't
zoom
get last
into
time.
Please.
Okay,
It's all right. Okay.
no problem.
Thank you.
Let's go over that way a little. I thought I saw something in the gloom, which may or may not be a cast. Yeah, what's that?
Oh, what is that?
Is it a coconut?
Quickly zoom.
I hope it's a coconut.
It's a coconut.
It's a coconut.
Oh my gosh.
And look how much life there is in there. We can't get a
Oh.
better image of that.
Oh, yeah.
What in the world?
I want to see a coconut crab come out of it. Okay.
That would be too much.
Yeah.
Wow. Who lives in a coconut under the sea?
Try and get a
Little
great pic of that one,
stars.
please. I think people will love to see that. You can see worms are eating into the shell and everything.
Yeah.
Oh, yeah. That's right. Just give it a sec. It'll clear.
Well, that is just really discombobulating.
And go wide, please.
Okay.
To see something from land, it's so deep in the middle of the ocean,
Yeah.
isn't it?
Yeah, you know what I noticed? It's a bit hard to tell. It looked like some of the husk was left on top of it. So it could have been a natural coconut fall that somehow got breached while it was at sea. And the top hadn't really been cut off properly, so I don't think someone drank the coconut, threw it off a vaca or something.
Okay.
I'm pretty sure it was just one that drifted out.
A little urchin up there.
Yeah, let's go for it. Okay.
Zoom in on that, please.
Nice.
Thank you. Yeah, it's interesting because obviously we saw one piece of rubbish on a prior dive, but I'd say that's the first coconut we've seen as well.
Yeah, I think so.
And there are millions of coconuts drifting around in the ocean. But I guess most of them don't sink to the bottom. They've been designed not to do that.
Speaking of coconuts, we have a Canadian in the room. I don't know why I said that.
She's talking about you or me?
Wait, we have two Canadians in the room.
It's definitely been a long cruise, Maddie.
We have
Josh comes back with 3.
We have a couple of coconuts in the room. Tim, what are you most excited about when you go home to Jana's losing it over there?
Plucking coconuts off the trees in his native Canada, obviously.
Yeah. Lately, I think I've been dreaming about sleeping in a full-size bed when I get home.
That would be nice.
But yeah, no, it's first trip here on Nautilus. It's been great. This great crew, and everyone's very helpful, and really, really cool setup here. Yeah, so I've really been enjoying my time out here.
Yeah, it's been a really incredible cruise. It's funny how...
We want the zoom on this one for
Yeah,
sure.
let's
Okay,
zoom on
I'll
this.
set down.
how quickly
Okay,
it
go for
goes
zoom.
and also how slowly it goes at the same time. Oh, wow.
Oh, it's another one of those little tunicates.
Yeah,
I'll
on
just tilt up
Beautiful.
briefly.
a sponge stalk. Is that what we're seeing?
Looks like it. Ugh, how gorgeous. The shallow water, those things are called sea squirts, which I think is such a cute little name. Tunicate.
Okay, good for that. Thank you.
Very neat. Thanks, guys.
All right, so we heard from Coconut 1, Coconut 2. What are you excited about?
No, it's swimming. Don't worry about that. They'll lay that
I
last
think
one.
being able to walk for more than 50 feet in any given direction without having to step over something, duck under something in a straight line, maybe in different clothes too, would be nice.
Oh, yeah, like a new
We could
style
just
change.
swim over that.
Just different shoes, yeah.
Different shoes. Tall people problems over here.
Amen.
Yeah.
There's a crinoid
And my
over there,
family, of
and a
course.
sponge, I think.
It's your family, suppose.
What do you reckon, Sandy? You a little bit of zoom, please?
Yeah, go for zoom.
Yeah, that's one of those.
Ah, cute little sponge.
Yeah, thank you.
Do you want to see this one off to the left?
No, we're good
Okay.
with that,
That
I
looks
think.
like a little stuck crinoid.
Well, I'm presuming Sandy's good with it.
Thank you.
Let's continue. Maybe we'll head over that one as well. Yeah, it looks like another crinoid again.
Off in the distance there.
Yeah.
Yeah.
There we go.
There we go.
I hate these
No problem.
chairs.
And Ryan, of course, is flying Atalanta right now.
Yep,
First time on Nautilus.
that's right.
Hopefully not the last.
Yeah. Thank you.
Been really fun to sail with you. And to Ryan's left, we have Jaina. Jaina, you're traveling actually when we get off the cruise, but so I suppose, like, what are you most excited about when you're back on land?
I'm excited for fruits. When I get home, I'm excited for my couch and my cat and reality TV. I'm excited to sit on the couch, all day with my cat and watch reality TV and catch up as soon as I'm back.
Oh my gosh, just like one full week of rotting.
Yeah.
I feel that. I'm sure your cat is equally as excited to see you.
I hope so.
I bet it is. What is your cat's name, Jenna?
Can we get a quick zoom on that?
Her name is Riri.
Aw, Remy. Looks like a little stalk
I think
crinoid.
there's a few of those
On
out there.
On the stalk or off to the left?
No, there's stuff here.
Okay.
I think the other leg was looking at some of these as well. There's a few of them about.
Can you zoom in? Are you
Nice.
looking at the white thing there? Or?
No.
I think
Brown
the
on it.
kind
There
of
it is.
matted stuff up to
There's
the
Yeah,
a
left.
little
okay.
clump of sediment up there.
There's lots of small stalks, eh?
Weird.
Okay. So you've got this stuff here, you've got the stalks, and then you've got that one. Interesting. Okay, thank you.
Did you see that massive sponge structure that one of the other watches saw? Mikhail adjusted one of the photos of it, and it was really big. How big was it, would you say? Did you have lasers on it?
I don't know. I think it was like a couple meters at least
Wow.
wide.
Was
Really?
that on top of the seamount or on the planes here?
It was on top of the seamount,
Oh,
approaching like 2,000
Yeah,
That's
meters.
really
Yeah.
There was so much up there.
Just try and get up to the top of Atalanta screen again. Buy us some stop time if we need it. Of course, stop me if you see anything really interesting.
Yeah, I think so far we can do them on the fly. Hey, Sandy. So got a bit of space. Shall we zoom in on that little white fleck?
Oh. Okay, go for zoom. On the one up at the top there a little.
Yeah, up there. That tiny thing. There's a small sponge, is it?
That one there?
Yep.
Yeah, let's get a close-up on
Yeah.
that. Oh, it looks like a little Holothurian.
Yeah, I think so.
I'll tilt a little
tiny little trail around those nodules.
Oh, I
It's
didn't
been
even
cleaned up.
notice
Yeah.
that. Yeah.
That's epic, isn't it?
Well,
All right, thank you.
let's put a spit polish on those things.
Should have left my shoes down there. Come
Yeah.
back and pick them up later. You want to go for another small one while we've got the space?
Where are we looking? Oh, right over there, yeah.
Sandy, a couple folks suggested maybe that tunicate
Okay,
that you saw
zoom
might be a
in.
benthic ctenophore.
Could
Did you see
be.
that? Yeah.
We're definitely going into macrofauna land here. Another little gastropod, I think.
Yeah.
Yeah,
And then
yeah,
there's
yeah.
those
Sweet.
very small white specks, aren't there?
Good on you, thank you.
Yeah, that could be maybe bits of marine snow falling down.
Mm.
Could be really, really tiny little animals.
Thought I saw like a little tube protruding just above that gastropod just now. Like we were staring right down the barrel of it,
Yeah.
just coming out of the sediment.
Maybe just make a note and someone can check.
I think 5197 is the deepest I've seen our little Hercules at, right? I
Yep.
mean, I don't know if one of the other watches saw deeper, but
Yeah, no, we are eagerly awaiting our, if you're into arbitrary round number milestones, we're 5,200 would be a new little Herc record, which we ought to get a chance to hit coming up in the next hour or so.
cool. I was on Hercules' 1000th dive, and that was a cause for celebration. I love these milestones. They're so fun.
Which was the 1000th? Oh, of course.
Yeah.
8 dives ago, yeah.
What do we have there?
Oh, there he goes.
One of our fish.
Yeah.
Looks like it's moving.
Can zoom there.
Staring right at you.
Nice.
Oh, good pick.
Cool, great. Great capture.
Thank you. While you're there, do you want to look at that stem? Sandy, can you look at the stem right there? Yeah, might as well.
It's just moving so slowly, isn't it? It's just puttering along.
That critter on there is not doing a good job of keeping his stem clean. Thank you.
It's going to get an eviction notice.
Now that's a big tooth. I don't know if you saw that. It's huge. That was longer than your hand.
Wow.
You get them sometimes. They're not like an equal triangle. They're more, what's that, isosceles or sort kind of shape?
Is that just due to erosion or a different species?
Maybe a different species, yeah.
Yeah.
Really long teeth, it's weird. I haven't seen them very often. I think they're teeth, but we see them kind of at the last second.
One of the publications I worked for,
You want to go for that swimmer? What do you think our chances are?
Yep.
we were a science publication and we did really incredible holiday parties, and I won a megalodon tooth in the gift exchange.
Oh, awesome.
It was so cool. I still have it on my desk. Do you have
Good
any
for Zoom,
teeth?
Tim?
Yeah, I've brought two on the boat we can
You
look
do?
at during
Oh,
the show
yeah.
and tell. One from the QCed, one from here. Wow, look at that.
Oh, wow.
It's like it's walking on water.
What is that? I think you're off SPL,
It's
Sandy.
Remember that breaststroke isopod we saw
Yeah.
last night?
Yeah.
It looks the same thing.
Great.
All those
Oh.
legs.
I think it's right inside close to the camera.
That's great. Thank you. We're good.
Thank you. That was really pretty.
Let's go make some hay.
Do you remember what the scientific name of the breaststroke isopod was?
Munnopsid?
Oh, yeah, that's right. Yep.
Munnopsid?
Holothurian there.
Yeah.
I feel like the nodules are getting a little more spaced out here, John.
Yeah,
You
could
agree?
be, eh?
Looks like it.
I think it's more medium now, eh? Let's
Is
get
that
to a
a
geonon.
sponge just there? Yeah, it is.
Oh, there's another one of those long teeth. Let's just see if we can fly over it. And just let us know if you want to zoom, Sandy.
Just fly over this, or did you want to stop and zoom?
Let's just fly over this one.
Okay. I'll go off to the side of it so I don't wipe it out.
A pretty little sponge.
Sorry, can we... sorry. It's one of those long teeth, but again, I left it too late. There's another one, I
Right.
think.
something emerging up ahead. You got some targets on the sonars, yeah.
Yeah. It's like a mound or a rock or something. Oh, that's interesting. You can see we have a small lip in
Yeah,
the
let's
sediment.
go over to those rocks there.
They might be a little bit outside of our range, but we'll get over towards there.
Gene, a little more tether there, Tim, or is that all right?
I think we're okay there. Yeah, you're
Weird
12 meters
change.
off seabed there.
It's a big mound, isn't it?
Yeah, and then there's something behind it. Can we go up and see that too? Possibly.
Yeah, quite a bit, either larger rocks or larger nodules here, isn't it?
Yeah, there was a definite change. It was a little
Yeah.
depression.
Big
Yeah,
mound of
if you look
irregular,
at
big
look at the
mound.
Atalanta sonar, how much more backscatter there is on that.
Yeah.
That's because of the large pieces, I think.
Yep.
Can we get a zoom on that one?
Yeah, could we?
Okay.
And there's a sponge, it looks like, to the left as well, huh?
Yep.
Are you able to zoom on that?
Trash? Question mark?
It sure is.
Something.
Anthropogenic.
Trash piece
Wow,
number
water bottle.
Wow.
Let's go.
Okay, we're done with that one.
Trash. It's terrible.
2. Okay.
And a little sponge behind it. Life amid the waste.
Nothing living off on that like the coconut.
Nodules are-
5198 for depth, getting so close.
We're
It's another one
so
of that big
close.
mound.
It was like a little depression here, wasn't it?
Yeah.
Yeah, if we can go over the Holothurian, it's not too much trouble. Oh, look at that.
Yeah.
Like a piece of crust.
Looks like one of those sponges.
Yeah, that's just what it is, eh? Those big flat
Going to
ones.
quick zoom. Here, I'll tilt down.
Nice. Glass sponge.
You good with these, Sandy? Yeah?
Yep.
Is that to the left there? Another sponge or a Holothurian? Yeah.
Can we zoom in on these? Let's go with this one
Okay,
first.
go for zoom.
Looks gray and crusty.
Yeah, it's just a big old rock or something.
Huh.
Maybe it was a piece of pumice originally. Thank you. Let's continue.
The geology is changing
another
again.
little
It really
depression
is,
coming
yeah.
up there.
Will we hit 52?
I don't know.
Oh, we're so close, 2 meters away.
We go for a little Herc burrow mode
I
to
would
play
like
the
to see
drill.
that. Not just our
Snuck that
Quite
on
a
there last
round nodule there, isn't it? Or bigger anyways.
Yeah.
Looks
Very
more like cannonballs.
irregular. And getting knocked in the head with one of those would hurt. Okay, let's see.
Yeah, if we can just go over those 2 features.
Lots of these little mounds.
Yeah. When you get into these irregular little depressions, there seems to be a lot more bioturbation, eh? Should we go for a zoom on this one?
Sure. Okay. Can you zoom in, please?
That's a little urchin and a holothurian. 2 echinoderms, but very different appearances.
We saw some of those last time, didn't we?
Yeah,
Last night.
wow.
Uh-huh.
Lots of spikes.
Thank you. You want to go for the Holothurian as well? Let's do that while we're here.
Holothurian.
Oh, and there's a shrimp over
Yeah,
there.
can you zoom there?
Shark tooth to the left, maybe?
Yeah,
Good size one.
yeah.
Oh, it's a little xenophyophore right there in the front.
Sweet. This one here. Yeah.
Oh, good eye, yeah.
Yeah.
And look at that. Oops.
Yep.
Can we zoom in?
Xenophyophore.
To the right there, yeah.
We'll do the
Xenophyophore.
double on that
Oh,
one.
look at that little baby there. Did you see it on the rock? Cool.
See if we can get a good photo.
Strange.
Thank you.
And wide,
Thank
please.
you.
Yeah.
Yeah, Holothurian.
It's like another crinoid that we passed over a moment ago.
Mm.
I know it looks like we're going really fast. We're quite close.
Mm-hmm. No, we can go that way, please. You want to zoom on that one, Sandy?
Yeah, let's zoom in on that.
Okay, zoom in.
Looks like a little sponge. Yep. That's exactly what it is.
A bit like a lily leaf, eh?
Mm-hmm. It does look like that.
Should we check this one out, Tink?
Yeah. Okay, go for zoom. I'll tilt up for you.
Nice. Thank you. I love those little speckles on the ends. They look so pretty.
Okay, thank you.
John, someone is curious about the geology of this area that we're in. Why are we seeing less nodules and more sediment? And how does that compare to previous areas we've been in?
We chose this area because we were expecting to find less nodules, but not necessarily at this point of the traverse, a bit later on, I think.
Do a quick zoom on the fly.
Big slabs of rock there on the left, yeah.
It's certainly very irregular.
Indeed.
And so the ferromanganese precipitates reflect the history of their growth, obviously. And when you get irregular things like this, it makes you think that the geochemical conditions themselves might have been irregular. Some of the larger fragments, though, I'm not completely sure if they're ferromanganese all the way through. Normally you'd take a little hammer and break them in half and have a look inside. Some of them might be bits of pumice or other bits of crust or something that have come apart over the eons. So yeah, the ocean seems to be very, very homogeneous, but it's not. It's just got small local variances, and over time that can manifest in different, we call them fascies of nodules. So let's go on that one, please.
Let's zoom on that too.
Okay, looks pretty. Maybe get a nice photo of that. So yeah, that's what you're seeing is slight differences in the formation. The two main candidates usually for changes in nodule fascies is the thickness and stability of the geochemical conditions that form them.
There we go.
It's a bit different, eh? Have you seen one of those before?
Yeah. Very pretty sponges.
Do you recall seeing that one before, that form?
Not that color at the bottom, no. It's a bit reddish, which is different from what we've been seeing before.
Okay. And go wide, please.
Okay. So here, for example, you can see there's a lot of slabs suddenly.
Sure.
It's likely that that was a crust that's either broken up or the nodules are being pulled together.
Okay. So it all comes back to how this landscape was formed.
Yeah.
In a lot of ways, I suppose, I should say.
We've noticed a couple, can we zoom in on that? Sorry, I know it's a bit of a rush. We've noticed a couple of times that there's like little embayments you can see. I'm just going to draw it on the screen. See that there? There's some micro relief here on the seabed, and we're not sure why that would be the case.
Zoom in a bit, please.
Yeah.
Oh, wow.
Look at that.
Send that to Brian and Ellie to have a look at.
Cool little creature. Nice.
When you get back, Sandy, he'd be a good guy to catch up with from Eckhart. He's the xenophyophore fella. Yeah, so that micro-relief might then reinforce with nodules and then reinforce again, yeah. We are a little far north now, but there's also the possibility of recent volcanism adding some components into the mix. We know that in Aitutaki, which is quite a way south still, several hundred kilometers, there's volcanism not so long ago. And yeah, maybe there's been other exotic things added to the mix. I think the previous shift were pretty sure they found some pumice on the seabed, which could float on the seabed and then sink.
Sure, yeah. Yeah, we saw pumice a couple days ago, didn't we?
Yeah.
Gosh, this depth is really just taunting us. 5198, we're so close. Hi, Lindsay.
Hey, Lindsay.
Can we go for zoom on that? It's all right.
Bouncing a little bit here.
Need a little more tether.
There, okay, go for zoom.
Aw, little brittle star. Very
So
pretty.
we saw that before. Is that rock standing on end?
Kind of looks like it, right?
Yeah, it does.
How on earth would that happen? That doesn't make any sense.
It's a strong starfish.
Yeah. That is really a puzzle. I don't know why a rock would look like that. I'm wondering.
Lindsay, are you joining our watch?
It's the best watch, you know.
Am I? Oh, I'm talking.
You're on.
Yeah, I
Welcome,
am.
Lindsay.
Yeah, my name is Lindsay. I'm a data engineer here at the Ocean Exploration Trust, and I am going to be shadowing Sandy as she data logs today.
Yay!
We are approaching the off season and thinking about making changes to some of our software and different technical processes that we work on. So I just want to see how Sandy is logging data and see how everyone else is using our different software to kind of get an understanding of maybe what could be changed or improved.
Well, that's very exciting. I'm very happy to have you here.
Yeah, this is my first time ever on SPL or on camera, so I'm a little nervous, but excited to be in the van.
We got you. I'm so happy because we were just talking about how we're the couple that never gets to see each other.
I know.
Lindsay and I are cabin mates, and so she has been working during the day, and I've been working that overnight late watch, and so we just have been kind of passing by each other as she's waking up and I'm going to bed, or vice versa.
Oh, that looks like a Holothurian there, doesn't it?
Yeah, you want to zoom?
If we just
We'll
fly a little closer to it, we can probably...
Zoom on the fly, please.
Okay. Okay, go for zoom.
Great, thank you.
Thanks, Tim. Thanks, Jana.
Yeah, so there's a couple of folks in the chat who are thinking about maybe what could have caused that rock to rotate up. One person is asking, could it have been in fauna?
Totally could have been in fauna. Obviously, it's hard to say if it's in fauna because they're in the ground usually, but who knows? It looks like it's been there for a long time because of like that strange like filamentous growth kind of like billowing out.
Yeah, we saw
next to
one,
it.
and we saw one further down earlier, so it's quite unusual, and then to see a couple like that so close is odd. Yeah.
Very strange, though. Very cool, very interesting. Leads to more questions. You know.
Yeah, another person asked kind of the same question, is it intra-sediment benthos, like worms? Yeah.
Maybe, probably.
I guess the only way to know would be if we were to see them in action.
Right.
Or
If they peek their little heads out or something.
yeah, come on, worms. Come hang out with the Gwoat. Greatest watch of all time, the Rolex.
Okay, zoom in, please.
No, yep, okay.
Oh, you okay?
Yeah, we're okay with that, but I am actually interested in the one over here.
Okay, and go wide.
We've got the space. That one there, is that in range? Oops.
Yep. The taller one there?
Yeah, that one.
Nice, look at that little guy. Oh yeah, let's zoom into that one straight ahead, that little oval shape.
Oh, there's a shrimp.
Yep.
Let's get down to seabed here.
Yeah, Lindsay, one thing that I've really appreciated about Nautilus is how
Okay, go for zoom.
multifaceted everyone's careers are, and a lot of folks can hop from the comm seat to the video engineer seat, data engineer, and
Look at that.
data logging, so it's cool.
It's interesting, there's a red pattern
Doubler?
on a lot of the nodules.
Yeah.
Oh yeah, okay, that's little sponge. Or same.
Carnivorous?
Yes.
Okay. It's really hard to see that. Good eye.
All right, when you zoom out, I don't know if it's in range. I'd like you to zoom to head over to this feature. That one there.
Zoom in quickly.
This one here, please. Could get panned down slightly. Okay, hold it.
And go wide, please. Thanks.
I don't know what that is.
Tim is
Love
really
it.
great. He'll zoom in on anything that you want. And if you can't, he'll let you know.
And then it's racing to get back into position, past all the teeth. And
You know, if I didn't know better, I'd say they're bones, but they dissolve at this depth.
Yeah.
So I'm just wondering if there's some cartilage or something from an old whale fall or something that's ended up in these strange orientations.
Huh.
Looks like that's another one of those little stalks that we've been seeing, yeah?
Which one? This one here? Or this one?
Yeah, no, the one right below us.
Oh, that one? Okay, you want to look at that one?
Yep, that's a little stalked
Are you happy?
crinoid. Yes, I am.
She's
That's an
happy.
echinoderm.
And do you want to go for the sponge up here as well, the vase?
Yeah, let's go up there.
that's probably a little too far away to zoom on. Oh yeah, especially with that dust cloud.
Okay, if we could just hit maybe a zoom.
Yeah, I'll get a bit closer to it.
Thanks, Tim.
John, you said earlier that bones would dissolve at this depth.
Zoom on the fly there. Okay.
Yeah, well, we don't know 100% for sure because the depth at which...
Nice.
That was a Holothurian, yeah. The depth at which we
cool.
have dissolution of bones varies in different parts of the ocean. But as I think most people know, the ocean contains quite a bit of CO2. And the CO2 is slightly acidic, certainly at the surface. We've heard of ocean acidification and things like that. But as you get deeper, the pressure, the water pressure, makes it more acidic, makes it more active, basically.
Oh yeah, that's really something.
Yeah,
and,
it is.
at these depths, generally, you're below a...
Tim, could we get a zoom on the Holothurian, please?
Yep, go for zoom there.
Cool.
called the lysocline.
Observation
Below
bio.
there, it's much more acidic.
Beautiful. And then that's an echinoderm. Yep. And then Holothurian. Look at his little 2 feet spinning over.
Whoops, it happens.
Aw. Perfect, good job.
Back out. So yeah, that's why we think, well, we don't see much bones. We can look at the sediment as well. The sediment's not full of calcareous fragments. If you go and look at the sediment from the top of the seamount we went to, it's full of little white flecks, which are little bits of calcium carbonate or chalk that are part of the sediment.
Yeah, something we've seen a few times are whale bones that have been sort of coated in that manganese polymetallic.
Down here you shouldn't.
Not at this depths, but
Yeah.
at higher depths.
One thing that does persevere sometimes is what's called the septum, which is, you know, in your nose, there's a bit of cartilage. Yeah, that'll survive. That was a good crust.
Little, yeah.
Yeah, so some of those videos where we've seen those manganese,
Oh, look at that crust.
manganese encrusted whale bones and whale skulls, you can always check it out on the YouTube video or on the website.
Look at that. Very cool.
On sediment, eh? I should have mentioned that.
Do you want to log that
can we get this?
Holothurian there on the left?
Yeah, please.
Okay, zoom in,
Yep.
please.
That was an echinoderm. Ooh, look at that. Is that a Holothurian?
Yeah, it's not.
Let's
an
get a
interesting
little
shape.
closer on that, actually.
At least it's not a plastic bottle this time.
It does kind of look like that sponge that was near it, though, doesn't it?
Not sure that's a sponge.
That's definitely not a Holothurian. How weird is
It
that?
certainly looks gelatinous, and it has a little something on the inside there.
Okay, we've got a good image.
Mysterious cnidarian. Yeah, mystery blob, that's right. That there is a Holothurian on there.
Complete with the track.
We just passed over it. Oh, and then there's one there on the left, if you want to log that one, yeah.
Okay.
I think these are the sea pig variety of Holothurians, which is quite a funny name. And then there's this little stalk. I wonder if it's maybe we're a bit too close to zoom in on that.
Yeah, I feel like manatees being called sea cows kind of makes sense to me. The sea
Yeah.
pigs, I don't understand.
I know.
Does it make sense to call it a sea cucumber?
Yeah,
Yeah.
cucumber shape.
Right? Yeah. Especially those shallower water ones. I feel like they have like the little lumpies, like a land cucumber.
Yeah.
These ones, the only thing I can think of sea pig is either they're eating a ton or like their front little papillae are like kind of like snout like.
Headless chicken makes so much sense to me. What about a sea pickle? If we have sea cucumbers, we should have pickles, right?
Just
That's a good point.
Evil Holothurian, a sea pickle.
Oh, a sour Holothurian.
Ooh.
Let's get up on that little white thing at the front.
I mean, I feel like if the ocean is more acidic down here, that's kind of pickle-ish.
Sour already,
I don't know, yeah.
Are all sea cucumbers sea pickles down here? Perhaps.
It always comes back to food.
Yeah, we might go for a zoom on that, please.
Yes, please, Tim and Jana and everybody. And then do observation bio, most likely.
Okay.
Once we zoom in, we'll see what it looks like.
And we might look at that rock on the right when you're done with that.
Okay, go for zoom.
Polychaete.
Yep.
Ooh.
That's a big
pretty.
polychaete.
Yeah.
10
So
centimeters,
that you're just
it
going
looks
to
like,
keep
huh?
other clicked because we don't
Okay.
have a drop down for that, but it's annelid, so ANN. Look at all those parapodia on the side.
That's
Wow.
beautiful.
Oh my gosh, they swim so gracefully.
It reminds me of Aladdin. And the
Yeah,
magic
like a flying
carpet.
carpet.
Yeah.
Yeah. And it looks glittery to me.
That's great, thank you. Can we look at the rock on the right hand side?
Beautiful.
The rock on
Yeah,
where?
so that's the little polychaete. It's a marine worm.
Just turn it
Cool.
slightly.
Yep.
Hopefully it's
Yeah.
still, there it is, this one here.
Yeah, we have, oh, here.
Okay.
Maybe this would be helpful.
Oh,
We have all
thank
of
you.
these, the
So
little
check
codes.
it's probably a prime candidate to be a bit
Nice.
of
And then this is a
pumice.
little guide.
Okay,
Oh.
zoom if you can. Oh,
That's all right, just pan down a wee bit.
It's a strange little rock. What kind of a rock does that look like to you, John?
That looks like pumice.
Pumice?
Yeah,
Nice.
it's very much like
Is that a
pumice.
polychaete?
Oh,
That's
the
right.
polychaete's still hanging around. Yeah, no, that's great, thank you.
Aw. A swimmer.
Mm-hmm.
So, I think it's just a cast, but let's go check. There's so many odd things in this neck of the woods.
There's something out to the left too.
Yeah, that's
Kind of
another one
flat.
of the, oh, to our left, okay. That's another one of those,
Star.
yeah, oh, it's a star, yeah. We can just fly over it, I think, or would you want to zoom in, Sandy? Go for a zoom, please.
okay. Zoom away.
Interesting, it's on top of that little mound of sediment. I don't think they
Yeah.
make it, do they? It just happens to be there.
Unless it's just waiting for whatever's in there to come out.
Yeah. It's a bit like that lizardfish, you reckon?
Yeah, ambush predator.
Yeah. Beautiful, thank you.
Sandy, are there particular organisms that you're more interested in seeing than others? Like, would, if you, in that instance, there was maybe a sponge and a starfish, is there one that you would choose over the other to do a zoom in on?
Sorry, can we head over here?
Yeah, up to the right, Tim.
Give us a touch of zoom.
Anything that looks out of the ordinary, I would be most interested in. This looks like one of those
Okay.
abyssopathes,
center
which
up
is a
and
type
you
of
can zoom.
You
cnidarian.
happy with a quick
I would
zoom,
love
eh,
to-
Sandy?
Yeah, I would love
Yeah.
to get a zoom in on that.
Sorry. Meant to tilt up and I picked up and said, okay, go for zoom.
Oh yeah, we've definitely seen that before, huh?
Yep. Okay, I think, yeah, we got a good ID, thank you. We got such a great picture of that on the previous shift, didn't we?
Yeah, we really did.
Okay, head over that way.
Oh, I might have to miss that one.
No, it's okay, just head over.
Getting ahead.
It's fine. I think it's Holothurian. Yep. And another Holothurian, I think, on the port side. Okay, give us a quick zoom while we're here now.
Okay, go for zoom. Oh,
Yep. Okay, you got the one there, Sandy. All right, thank you. Zoom out and continue. You head over that thing, please. What do you reckon, Sandy? One of those sponges?
Yeah, I would say so, but we can swim low over it and just take a gander.
Do a quick zoom, please.
Okay, go for zoom.
Yeah, that's what it is. Thank you.
What do you think that is?
It's the sponges we've been seeing, they've kind of got like a crenulated look to them.
Oh,
They're
okay.
always on the seabed without a stem. We've been seeing them the whole trip. And continue that way. Oh yeah, it's a little crinoid. Sorry, can we get a zoom on that, please?
Yep.
Is that too late?
I think I can.
Just a thing there. And that's a bit late.
Let's see.
Beautiful, thank you.
Okay, can you zoom on that, Jana?
That's a big tooth.
Oh, wow.
Oh,
That's
Another tooth,
got
yeah.
to be 1, 2, 3, 4, 5, maybe 6, 18 centimeters.
Wow.
That's two-thirds of a foot long.
Oh my gosh.
Do you think it's like fossilized?
Oh, it's an ancient shark tooth coated in manganese. You see the lasers, right? They're 10 centimeters across, so it's,
Yeah.
with the perspective is longer. We can go now, thank you.
Okay.
Yeah. If we see a fossil, do we count that as a biological observation or geological?
Oh, they're more like curiosities, the teeth at this stage. I didn't even make
Yeah.
an observation.
We've seen a lot so far on this trip, and just on this cruise in general.
Yeah.
Lots and lots and lots.
Nothing else survives, and so that's called relative accumulation, basically, because everything else gets dissolved. It's just your teeth and a small bone in your ear.
Your
Look that up.
otolith.
What was it called?
Otolith?
A otolith, was it? Yeah.
Yeah.
And maybe a little bit of cartilage survives, yeah.
Yeah, I guess I've heard that the bottom of the ocean like traps a lot of carbon in it. Is that kind of what makes it more acidic down here earlier, what you were saying?
The amount of carbon uptake in the seabed itself at these depths isn't that high.
Okay.
However, if you go to a place above what's called the lysocline, where you get chalk accumulating, that's the single largest carbon sink, if you like, on the whole planet.
Wow.
That tends to be closer to the equator, and it tends to be in the order of where the seabed is about closer to 4 kilometers, not 5.
Okay.
So down here, the reason it's acidic is because of the CO2 in the water itself. So CO2 is a mild acid, and let's say you had 2 bits of water with the same amount of CO2 in it, but there are different pressures. This is at the pressure of 5,000 meters, and the other one says atmospheric pressure. This would be much more acidic, even though it's got exactly the same amount of CO2, because the pressure forces that CO2 to be more active in the way it attacks. So it's effective acidity. I don't know if the pH would be any different, but the effective acidity would be quite different. That's
cool.
Yeah.
And we've talked a lot about that CCD
Can we get a look
too.
at that one there? Thanks.
Right, the CCD.
Yeah, the calcite compensation depth is the depth where you get very little calcium carbonate remaining. I think it's, I should go and check this. I think it was about 5%, but I'm not sure if people even agree. So it's where you don't have zero,
zoom,
but
please.
very little. So that's a few hundred meters below the lysocline. So it's at different
And
depths
you
in
can
different
zoom in
parts
there,
of the
Jana?
ocean.
Looks like one of those, similar to what we've seen
Yeah.
before, one of those carnivorous sponge, so that's going to be...
Tilt up a
Thank
bit
you.
for you. Okay.
Move on.
And wide, thank you.
Anyway, above the lysocline you get a rock called chalk, and you'll probably be familiar with that because the White Cliffs of Dover are made of chalk. It's a rock made out of, it's like a limestone, but it's made of very, very fine microorganisms. The fustules, or the skeletons, are the microorganisms. Below the CCD, then you get these red They're called red muds. Actually, they're browny, like a chocolate brown colour, maybe the hint of red. And they're made of mineral clays. So that's how people have worked out what I said before, basically, is by going around the world and mapping what's on the seabed and then looking at the chemistry and realising that at a certain depth you no longer accumulate limestone. No, there's no reefs or anything like that. There are still calcite-based life forms at these depths. But yeah, I think
for that, Tim.
we're good with that, Sandy? Yep. Okay. But they have a little extra pressure on them because of the acidity of the water.
As a casual observer, it seems like more of the kind of structured animals that we're seeing down here tend to be those glass sponges. Is that because, like you're saying, the calcium carbonate can't really form?
Yep, that's right.
Cool. And that's also why, like, the black corals and stuff don't use calcium carbonate as their exoskeletons.
Ooh, I didn't know that.
Yeah, they have an internal one made of, like, a chitinous material. It's pretty cool, yeah.
Cool.
The chemistry changes, everything changes.
And the nodule chemistry changes. So
Yeah.
calcium carbonate can accumulate metals. It's a version of limestone called dolomite. And if you can't have calcium carbonate, then the metals are liberated. And one place they can go into are polymetallic nodules, into the manganese structures. So when you get nodules at all depths, and we saw that on the seamount, even at the top of the seamount there were nodules. But it'd be really cool to check the chemistry at each depth one day, so we could see how much calcium or how much silica is being found, or how much manganese, iron and cobalt and nickel accumulate at each depth.
Yeah, on the previous cruise I was on, which was NA-174 in the Marshall Islands, our geologist, Dr. Val Finlayson, was able to see some calcium carbonate in rock samples that we collected by shining like a UV light on them.
Oh, okay.
That was pretty cool.
Yeah, what depths was that at?
That was shallower than this. It was Big Herc.
Yeah.
I don't remember, maybe like 2,000 meters? Yeah. I could go back and check later though.
Did you collect rocks too and do rock cutting on the back?
Yeah, we did. I didn't use the rock saw. I was too afraid, but I saw like Val was doing it and a bunch of our
go for
interns.
a zoom on that stalk there, Sandy?
Yeah, let's do that.
Thank you.
Right there. Okay, go for a zoom.
Yeah, 2 kilometers is well above the CCD pretty much everywhere. There you go. We can get it.
Is there a chance we could double that?
Yeah.
Cool.
That's great, thank you. You good with that one, Sandy?
Yep, thank you.
Thanks. As you come back, there's a secondary down here. Keep zooming out. And it's down below here, this thing there. Can we get that?
Okay.
Okay.
Okay,
looks
zoom
like a
in there, please.
xenophyophore there to me.
Mysterious brown object. I
Yeah,
thought it
or
looked like a tube there.
mysterious.
It's not a tube.
Kind of does, right.
A rod. Thank you.
Do you think it's dead if it's covered in sediment like that?
I don't know. That's why you have to ask a specialist.
Okay. Yeah.
So there's a lot of, probably the most populous animal down there is a thing called a xenophyophore, and there are people who specialise in those and who have collected samples, are familiar with it, hundreds of different forms that you get. There's probably as many types of xenophyophore as there are of all the other animals put together.
Wow.
Wow.
And they can sometimes confidently say what they think it is. Sometimes they say, I've never seen that form before. So we took some samples last year, part of the Women in Science expedition that we ran here in the Cook Islands. And that was the first semi-systematic sampling for xenophyophores ever done. And the specialist who's looking at that as part of the team is Dr. Brian O'Malley from Eckhart College in South Florida. And he's already found a few forms that he's never seen in his career. So they might even be endemic to the Cooks, we're not sure yet.
Cool. We have a question from the chat. How do people contact OET for ocean surveys? And that's a really great question. Typically in an expedition season, it takes many months or years to put together the expedition and the cruises and the different partners based on the objectives and where we'll be operating. But John, this expedition was put together kind of at the last minute. So were you a part of that planning process and bringing it all together?
Yeah, I think this was basically pulled together in 3 weeks. And really hats off to the NOAA and OET folks who were charged to add an extra cruise into their annual schedule. Yeah, certainly it was not a especially calm period when you're pulling together
Very
something
busy,
like this.
yeah.
And even today we're still sometimes adapting things on the fly because we only had a few hours to make certain decisions.
Yeah.
And no chance to really do a careful data review. But no, it's been a great success so far, this cruise. We had a few setbacks at the beginning, but to do with weather.
Yeah.
So no, and the planning process, yeah, I think that's how you see a team work together well, and this team's really pulled together in a couple of key ways, very professionally.
Oh, yeah.
Firstly on the planning and putting it together, and then secondly when we had those setbacks again, there was perseverance and calm and determination from all members of the crew. And the outcome of that is we're at almost 5,200 meters and an exploring part of the seabed that's never ever been looked at before, which is no trivial feat.
Absolutely not. Especially when we spent the better part of the beginning of the cruise weathered out. We really came back and sent it. Three 36-hour cruises back to back to back, right? Pretty impressive.
We've got a chance on that one. Yeah, I mean, that's what the team do, you know. So, and often when you start doing something you haven't done for a while,
go for zoom there.
You have a little bit of teething problems, but.
And then could we go over to the left after that, please?
Yeah,
thanks, Tim.
that's great. Thank you. And yeah, up here.
Oh yeah.
Was that previous one a Holothurian? Yeah. Oh, cool.
Someone else wants to know, are the nodules we're seeing only on the surface, or are there also some
Zoom
underneath the
please.
sediment?
The short answer is I don't really know, but in the past where we've done systematic sampling using a machine called a box core that can go 30 or 40 centimeters down, we've found about 3/4 of the nodules at the surface and roughly 1/4 at depth. There's been drilling, ocean drilling using a big drill ship that's found nodules down to 200 meters, but by and large,
and
most of them are at the
please.
surface, and a few might get buried in certain places. So the reason they're all at the surface, of course, is something that people speculate on. And the thing is, it's difficult. Any hypothesis you come up with is a little bit difficult to test because you can't really simulate it because it's such a slow process. Yeah. And one idea has been to try and do some mathematical modelling and see if it leads to supporting it, but that's also very complicated. And my leading hypothesis is that everything that happens at the seabed happens at a different speed. And there's like, I can't remember now, 9 or 12 orders of magnitude of speed. It's a massive range of speed. And the slowest thing of all is the growth of the nodules. So it's not like you grow nodules faster than sediment accumulates. Therefore, if the sediment's accumulating faster than nodules are growing, for example, something's keeping the nodules on top.
A bryozoan or something up to the right.
So get that right. And one likelihood is bioturbation, action of animals and things like that. So Holothurians picking up stuff and dropping it down again. And
just up past it there?
Right between your
lasers.
Yep.
Okay, zoom on that.
So that's kind of my leading hypothesis. But
looks like a bryozoan.
All right.
All right. And wide.
But there's been a lot of other ideas out there.
Sure.
My favourite one's the Brazil nut model.
What's that?
I don't think it's correct, but it's just such a cute idea. Well, you take a jar of nuts and shake it, and eventually all the Brazil nuts are at the top.
Huh.
I think it's just to do with the fact that the small ones slip between the big ones. And so the idea is that over millennia of earthquakes, all the mud's fallen in between the nodules. I don't think it's got a lot of credibility. For starters, there's no glass on the end of the jar, right? You're not shaking the nodules against something.
Yeah, right.
Yeah, but
It's a fun name too.
it's a great idea. It's another crinoid, I think. Now we know that currents are really busy, and we know that life is really busy, and we know that it's probably easier for sediment to fall off a nodule into the gap between two nodules than it is for it to fall up onto a nodule. And we know that worms that are digging through the infauna that's digging through the sediment is not going to burrow through a nodule, but it'll burrow through the sediment and take out its food and put it behind itself as it goes. So all of those things lead to that idea.
Sorry, I'm not really hearing you. Okay. Yep. Okay. Yep. No, not really. Yeah, because it seems to be correct in here, but yeah. Right.
I'll make a note of that in the logbook.
Okay.
Sandy, I have a question for you. Someone is observing that some people might look at this and think that there's very sparse life, right? We're talking about all this biodiversity and all these organisms that we're seeing, but when you look at it, maybe it kind of just looks like a nodule field. What's your take on that?
My take on that is, while there is less biomass than maybe some of the other biomes on this earth, you have to remember that this is a more extreme environment, and so the creatures down here are going to be living under extreme conditions, and so it's not as easy to get nutrients. But there's a remarkable, diverse array of life. Like, take a look at what we're looking right here, this cnidarian, this abyssopathes. This is a crazy looking organism, right? It's beautiful. And there's tons of other things we've been seeing, very small, you know, maybe not as big and flashy as, again, some of the things on land, but the ecosystem down here is quite remarkable and very precious and very cool to explore and continue to discover. The more we look for, the more we see. So we'll always be learning new things.
If you're taking a log, Ryan, can you also say that we're weighted more to port?
Okay.
I also think it's important to note in these surveys that the animals that we're seeing is definitely biased towards, you know, sessile, non-moving animals, and especially the big ones. So all of our ROV noise might scare away some other creatures. And obviously, if anything is microscopic, we're not going to be able to see it. That actually is a question I had for the ROV team. Do you guys feel like is Little Herc quieter than Big Herc? Like, do you think we scare stuff away a little bit less?
Yes, Little Herc is definitely quieter than Big Herc. Mainly, well, it's got a lot less mass to begin with, but it also has electric thrusters that go to 0 speed and are quite silent when they're not spinning, and then they get louder as they spin faster. But Big Herc has a hydraulic pump and hydraulically actuated thrusters, so the electric motor is constantly turning the hydraulic pump to give pressure when you need it. So yeah, it makes a lot more noise.
Huh.
Interesting. Thanks.
And another big difference is as soon as Big Herc is powered on, that hydraulic pump is running constantly.
I think we're good, Tim.
Okay.
Keep going.
So yeah, I know E.D. Witter was making a note of that, the noise difference between subsea vehicles. when she was trying to image the giant squids. And she definitely concluded that electric vehicles are able to not really sneak up on animals, but they seem to disturb all the animals a lot less. But that's a theory, you know. Seems accurate, but
Cool. And is Little Herc able to use like the electric thrusters instead because it's smaller, or just because it's newer?
The electric thrusters have been a lot more available in the last kind of 10 years. But yeah, being smaller makes it easier to put electric thrusters on too.
Okay.
And to talk to that point, the ROV is not the complete solution for understanding biodiversity. Normally animals are in 4 size classes, and those 4 size classes are a result of the sampling methods required to study them. So what we call megafauna are 2 centimeters and above, and normally photography is used for that. This particular camera is got such a powerful zoom that sometimes we see the size class below called macrofauna, but of course you can't do the whole view for that in one go. You've got to zoom in. And for macrofauna, the idea is you need to take physical samples now. You need to take multiple box scores and filter out all the macrofauna. And that also, of course, helps you with the infauna side of it. And then you go smaller again, you need multi-corers, and smaller again, you can start doing just eDNA and things like that, because it's bacteria by that stage. So marine biologists use all of those things. And then for some of the larger animals that might be shy of something like an ROV, you get a bit of a chance to look at those by using baited landers. So what you do is you put some bait out, and then it's completely quiet apart from the lights. And then usually over several hours, or even longer than 12 hours.
I wonder if we could get a zoom on one of those white flecks when we're coming up to another one, please.
Yeah, you see even the burrow. Is that a burrow or a shadow? So the biologists combine all those techniques to try and get a feel for what's on the seabed. Yeah.
Cool, thank you.
We have another question about how to measure nodule prevalence. If we don't have ROVs, how do we do that remotely? Sub-bottom profilers, multi-beam, side scan, and what's the best way to make maps of these nodule fields, given that information?
Yeah, that's a good question, because I've spent quite a bit of time trying to answer it. I don't have all the answers. So the first thing you can do is you don't need to know where every single nodule is necessarily. What you might want is a map at a certain scale, at a certain level of confidence, to do with what we call abundance. So abundance is the kilograms per square meter. Can we zoom in on that?
Yeah, could we get a zoom on that?
Yeah.
Kilograms per square meter of nodules in a particular area.
Go for zoom.
And so you could take samples even at several kilometers or even 10 kilometers distance.
Straighten up on it.
And
Maybe
you could use geostatistics to interpolate between them. And
very cool.
That's often used. If you want a really high precision, is that like that one we saw last year?
Yeah, what is that?
It's a little small one. You said it was a not tapeworm, you said
oh, acorn worm?
Acorn worm, yeah. But it's a smaller one.
Maybe it's an acorn worm, maybe it's a ribbon
yeah.
worm, because the head does not have a shape as an acorn worm does.
Okay.
So could be ribbon worm.
Anyway, so
cool, thanks, Andy.
you can use photos to estimate the abundance.
Go wide, please.
For really detailed work. And the way you do that is either you measure every single nodule to try and get an idea of each one's weight, because the density is consistent and you can assume you've got the other dimensions, or you can use fancier statistics involving the distribution of sizes. So if you can characterize the distribution of sizes in an area, then you can just use the descent coverage or to do it.
Okay, so there are various technologies to do so.
Yeah, and what you do is the geostatistician who does that work comes out of it with
Horizon lower left.
a degree of confidence. And that confidence is what you use to go forward.
Okay. Yes, please.
People have tried to use acoustic methods as well as optical and physical samples. That's still very controversial. I don't know if that's going to become mainstream. It might work in certain areas if you collect that acoustic data fairly carefully. For that, you normally use the backscatter response from a multi-beam system. Things like a side scan system have too much attenuation over the swath to really be reliable in terms of
Could we maybe zoom in on one more of
quantitative
those little
thing.
white flecks as we pass by another one?
Want to go for that one, maybe?
Yeah, let's.
Okay.
Sandy, when you're logging something that we're going to zoom in on, do you usually wait until we've zoomed, or do you do it as soon as we see it?
If it's something that we've seen many times before and it has like a recognizable shape like some of those Holothurians, it's quite easy to just, you know, sort of recognize it. But if it's something strange or unusual like these little white things, I usually try to wait until we've got a closer up view before I try and make any guesses. Like this looks like another one of those little polychaetes, and so that's making me wonder if all of these little white specks we've been passing are small creatures such as these.
Everyone I've looked at's been a little bit different, haven't we? Tetrapods. And you want to look at this one as well? Oh, too late. That's okay.
I've got to get out,
No,
moving ahead.
Grab yourself some real estate. We can have a look at another one. Yeah, again, normally this technique is used for what we call megafauna, which is 2 centimeters and above. So if you look at the rounder nodules, roughly that size or half that size, but we're often going to things that are much smaller.
That's something that's wild to me, is this concept of megafauna. It really is like, what did you say, 6? How many centimeters? Two?
Yeah, a bit under an inch.
Bit under an inch, and then does that encompass like all megafauna beyond that?
Yeah, from something that's a bit under an inch all the way to a blue whale would all be considered megafauna.
It's a massive designation, isn't it?
Well, we don't need 5. I think 4's plenty.
What about the like super
white
megafauna?
Front, please?
Yeah. The one further ahead of the lasers?
Yes, please.
Okay.
And people have changed the definition to, some people use 4 centimeters, so an inch and a half. Some people use 1 centimeter, so less than half an inch. But the International Seabed Authority seems to have settled on the 2 centimeters or a bit under 1 inch. So that's what we tend to use here in the cooks.
Can you zoom there?
Sea urchin?
Great, Tim, thank you.
Yeah. Do you want to look at this little, oh, okay. Why don't we look at that quickly? Over here. I'm not sure. Could be one of those tetrapods.
Yeah, then we have those strange
There's
little...
one there, and that could be the same just
like
behind the rock.
That's a creature coming out of the sediment there.
Yeah, okay.
So as you're taking all of these observations and logging them, what do you do with that information after a cruise?
We have specialists that will come and rewatch the entire video, going to some of the places that we have logged and making more observations, more detailed observations and summaries. But we use these logs and these observations to compile different reports and summaries to give to different organizations so that we can better understand what we're looking at and characterizing the seafloor.
Yeah, that'd be a great one. So look at that.
So you basically take kind of a rough draft of the dive, and then there's another person that comes in and...
Gastropod?
Yeah, I think so. Oh, no, it's a little,
like,
what do you call it?
Oh, no, it is attached.
Like a
the mollusks? Or no?
We found the big ones a couple of days ago. A barnacle.
Oh, yeah, barnacle.
Yeah, thank you. Yeah, so we can use it to try and estimate at first the biodiversity, and we can even do some modeling to see how much, we can kind of predict how much there might be from how quickly we're finding new things as well. And then we can try and break that down by different zones and areas to see are we seeing differences in the biodiversity between different areas. We already know that we will find some differences. So, for example, at the top of the seamount was completely different animals to what are at the bottom.
Sure.
But in these different parts of the abyssal plain, it might be a bit more subtle. And there's various statistical tests that people can do to try and understand how things are changing, either on distance or types of substrate. And these all feed into a process called marine spatial planning, And you need marine spatial planning to make sure that all the different habitat types are either protected or conserved in some manner, if any development were to be considered. But yes, you can't do all this sitting at a desk. You've got
No,
to go
absolutely
collect
not.
the raw data, and a lot. That's why these traverses are quite long. We need to see the same thing hundreds of times, really, to be sure that we're getting a
Could we get that
representative
thing
view.
up on the center right, please?
That one there? Was that it? Sandy? Yeah.
Oh, a shrimp?
Zoom quickly.
Or a fish? No.
No, it's a little...
That same Munnopsid, but on the ground.
What is it?
It looks like that thing that was swimming through the air, the breaststroke isopod, but
Oh,
this is
yeah,
on the
yeah.
ground because of all the legs trailing behind it.
Okay, okay.
It seems like a lot of the creatures down here are really long, if that makes sense. You know,
Yeah.
like there's all the cusk eels that are so long and skinny, and there's this isopod we just saw with the really long legs. Is that, I don't know, is that true? Is that like a pattern? I don't know. Because I would kind of think, I don't know, I feel like if I was in a really high pressure environment, I would want to minimize my surface area to volume ratio, you know? And it feels like they're doing the opposite.
well, they have...
That's a really cool observation, Lindsay.
they have equilibrated to the pressure, so I don't know if it's a pressure thing. But yeah, maybe by being long... Because remember, it's dark, and they often have very long antennae, so they can probably search a lot better in the dark with long antennae.
Cool.
Yeah.
Can you zoom on that too?
Yes, let's.
There you go. That's a great view on these things. We don't...
Tilt up here. There we go. And go wide, please. Thank you.
Sometimes I feel like we're in one of those like crime shows where someone is like, enhance, you know, and they zoom in. It's cool when you guys do that.
I think we're heading towards one of our better chances to go below 5.2 over the next hour or so.
You think we're going to make it?
Looking at the contours, there's a couple of points here that are at 5.2,
Yeah.
so...
I hope so.
And that smoothed that data a little bit, so...
Do you want to see this one?
Thanks, Tim.
Did any of you ever see that animated cartoon, Magic School Bus?
Yes, Miss Frizzell is my
Classic.
icon.
Love that.
What's reminding you of that?
I don't know, just the tiny little things here and looking so closely at them.
Well,
It's like that episode where they go inside Carlos.
Yeah,
That's-
exactly. That you remembered his name is cracking me up. Nice one.
So,
you want to go and look at this?
yeah.
Cool.
I feel like Magic School Bus just inspired an entire generation of explorers.
We'll just get a zoom on the way over, I think.
Okay. You can Zoom there, Jana.
Sea urchin. Well, yeah, Lindsay, you said all the like long creatures, but now I feel like we're just seeing all these like little round creatures.
I know, I cursed it.
The internet, the ocean wants to always prove us wrong. The internet too, I suppose.
My favorite Magic School Bus episode is do you guys remember the chocolate one?
I don't think
love
so.
that one. Yeah.
They like
let's
learn
go for
about
this one,
food webs and stuff. It's cool.
Okay.
That would have been a really fun show to write for. Yeah.
Okay, go for zoom.
Oh yeah, we can see them. Those are everywhere, aren't they?
Yeah, they sure are.
Thank you.
What was the teacher's name in Magic School Bus?
Miss Frizzell.
Oh, that's right.
I had a teacher in 6th grade, my 6th grade science teacher, and she loved ocean science, of course. Maybe that's where like it comes from, but she would always say dinoflagellates, like dinoflagellates.
That'd be cool if she was listening in and heard you talk about her.
Yeah, that was a while. That was a long time ago.
Can you zoom on that
Which one?
the
This one?
swimmer?
On the swimmer. This one down here?
Yeah, if you guys want it, that's
fine.
Polychaete, maybe? Oh,
Yeah, thank
no,
you.
that isopod.
Did you have something up higher there, John?
Oh, there's a couple of white flecks. Look, I think the one on the left is more. Oh, it's a bryozoan. Let's go the one on the left.
Okay.
Oh, okay.
Yeah. You good with that?
I'll settle down. Can
I
you
was
zoom
thinking maybe
in?
that stalk in the foreground, but
Tilting up a little.
Got a Holothurian. Do you want to look at this one on the left here?
yeah, let's.
Yeah. That's another one, isn't it? Or was it?
It almost looks like an anemone and a sponge.
Okay, thank you. If you zoom back out, there's a small thing we can look at while we're here without moving. Just pan down. That thing there.
And right there, okay. Okay.
Oops. All right, thank you.
Yep.
Have you guys seen the new sea cucumber highlight video on the website?
No, I haven't.
Oh, what? All of our favorite Holothurians.
From this particular cruise or?
Yeah.
Oh wow,
Yeah.
that'll be cool. It's my favorite animal, as you know, so
Yeah.
I'll definitely want to check that out.
I just saw it pop up today, I think. I wonder if our pooping sea cucumber made it.
I hope so.
I hope so too.
That was a highlight.
Stuff is the highlight of my entire cruise.
I hope did you highlight it and give it a 5?
Oh, I think I did.
You might be the person they're complaining about. Someone apparently has been highlighting all the Holothurians.
It was me. I'm the one. I'm the problem.
Well, they made a highlight video, so that means it was not in vain.
Could we get a
Gonna
zoom
do
on that thing there?
This one here?
Put on the
over right
brakes.
below us. That's okay.
That's right.
You're all right. It's okay. It's okay.
It's coming down.
I silted that one out. The tilt and the verticals are inversed, and I inversed it in my head there a minute ago. That's why we drifted off the seabed.
No worries.
It's okay. Thank you, Tim.
As a data engineer, most of my work is on the computer, but sometimes if...
Can we zoom in on that?
This one, yep. Center up here. Okay, go for zoom.
That's another one of those worms, really. It's got a long tail full of sediment. That's nice because you can even see the casts that makes. All right, thank you.
But yeah, sometimes I take my laptop up to the lounge and like watch this stream while I'm working. And I think I happened to look up just as the sea cucumber was pooping the other day.
You
so,
did?
Yeah, got to see that.
Yay!
It was weird.
I mean, okay, yeah, I'm like way too excited about it, but I think it's cool because we spent so much time being like, what are these mysterious tracks in the sea? And what are these mysterious deposits? And then we had both of our answers. The answer is always Holothurian. Oh, yep. Spoiler alert, the pooping sea cucumber made the clip.
That's great.
Yeah.
Well, it's actually talking a little bit about this platform because I did a survey in the Central Pacific a few years ago, almost 10 years ago now, and we took 20,000 photographs, and only twice did we find the Holothurian on the end of the track.
Really?
Yeah, and there was a towed camera system. With this particular system, because we can drive around and navigate in real time, we've seen that. It must have been at least 10 times now.
Could
maybe
we
20.
grab that little white thing in
This
the
one
center,
here?
please? Yeah.
So, no, this is certainly giving us a lot of insights into how things work.
Okay. Okay, zoom, please. Oh,
Crinoid.
sorry, I'll settle down.
Something up to that.
Oh, I've stolen all your light from you.
That's a pretty good photo. Thank you.
And let's get you back to port a little bit, Tim.
Yeah.
So Sandy, did you know like what a lot of these animals were or like how to identify them
We have
before
room to
this
go for
cruise?
that?
I knew some of them from different places that I've learned, but I had to study a lot of these before really starting the log. And a lot of
zoom
people
in,
around me know
please.
more than I do, so it's been cool to learn from them.
You good with that, Sandy?
Yes, Sandy, do you get like a study guide before you
Thank you.
before you sail?
No,
Specific
you have
to
to
a
make it your
region?
own.
You do?
Yeah.
We've got lots of room, haven't we? So she'll go for another little white fleck when you get a chance. pick whichever one you want, or that one.
This one in, okay.
Honestly, we don't know which one to choose. Any of them would be good. But
okay, zoom there. Oh. That one's hiding behind a rock. Can you go a little?
No, it's down below here. Down a bit more.
Okay. Tilt down.
There, I think it was that one. It looks pretty buried, eh? Oh, yes, one of those pteropods, or what did you call it? Matt, you gave it a name, wasn't it? Was it a pteropod or tetrapod?
Such an interesting shape, isn't it? Is it shelled?
And let's go wide.
Thank you. I don't think anyone's given us a suggestion yet.
Oh yeah, I think we saw one of those a little bit ago, and I just marked it down as like mystery flat diamond creature.
Oh, here we go.
Cool.
Is this what
Oh, pteropod.
Asako is suggesting? A pteropod shell. That could be it.
Thank you.
So we have another question from the chat. Obviously, these depths, we are at 5196, just waiting to get to 5,200 meters, but there's an enormous amount of atmospheric pressure down here, and someone wants to know why these organisms aren't getting crushed.
So it's a bit like when you swim into the pool and you dive down and you equalize your ears, but it's done on a massive scale, obviously. And it's like the equipment as well. There's different ways to have your equipment survive the pressure, and one is simply to put holes in it so the water can go in, and then what's around that equalizes. So these life are basically, and the rocks themselves are equalized to the pressure at these depths. Of course, it gets a little bit more complicated as you dive deeper and deeper into an organism structure. When you get down to things like proteins and things like that. Yeah, you got to go for that
Orangish thing.
Oh, the stem there? Yeah. But that's the simple reason.
Zoom in, please. Oh.
Huh.
I think it's a dead one.
A sponge or?
I don't know. Just, I'm not even sure if it was live or maybe a small piece of pumice. It was a bit round. Thank you. Let's go for that stem, shall we, Sandy?
Yeah, let's.
I think it's a crinoid, eh?
Okay, zoom there.
Yep, we're good with that, I think. Can we come back out? And let's head towards this fella.
Off in the distance there, or right just past this, that one.
Yep. You can just do a quick zoom on the way past.
Try, I get a little close to that.
Oh, you want to get a good one on that? Okay. You got enough time, eh? You can see it on Atalanta. Look at the white speck there.
Okay, zoom on that.
Looks like a glass sponge.
You good with that? Thank you.
Gonna get the tether out in front of Adelante again here so Ryan can see where we're going.
So Sandy, what I guess determines whether you're using this like paper book that you have versus like looking on one of these like NOAA identification guides?
This paper guide is pretty nice. I think TJ was one of the ones that curated it.
Let's go for the stem.
And it's cool because it has all of the ones typical to be found in this area in the Cooks. But if it's something that I've never seen before in this guide, or I feel like I know the name but I don't really know what it is, then I'll just look it up real quick.
Cool, thank you.
Can you zoom there?
Yeah, there it is.
Oh.
Thank you. I was wondering if that was one of those carnivorous sponges, you know, the ones that were like an oval with the cross things? It was just overgrown.
So what makes a sponge like carnivorous versus not? I don't know. I was under the impression that all sponges just kind of eat whatever filters through them, you know?
The carnivorous sponges in the deep sea are pretty cool, and it isn't typical. Most sponges are filter feeders, but these ones are interesting. They use their like spicules as little filamentous, and they pull them out and retract them and catch little crustaceans, which is pretty cool. The ones that are just, you know, filter feeders, they have all this like ossia in the osculum, so they pull the water in and out. But the carnivorous sponges are very unusual. I'm not sure exactly why they adapted, you know, or how they did,
Can we head over
but
here?
because of the nutrient dense, yeah, or nutrient, what?
Where was that?
Thank you.
Right over here.
Yeah, what's that?
There's not a lot of nutrients, and so they have to come and adapt in different ways. I wish I knew exactly why.
That's so
exactly
cool.
how.
Did not know any sponges could do that.
Right.
Okay, zoom there. Oh, I'm a little...
Holothurian, I think. Hey. That's great. Okay, do you want to go to that stalk that's just ahead? There it is. Get a zoom on the top of that, please.
Okay, zoom.
That looks like a hydroid. I'll log this one.
Okay, thanks. All right, we're good to go.
There we go.
Let's head over to that one. Should we try for one of those two stems?
Sure.
Sandy, do you have a preference?
Yeah, can we go to that long slim stalk, the
This one
carnivore
there?
sponge?
Yep.
Okay, zoom there. Okay, and I'll tilt up a little.
There you go. Great, thank you.